Now let's explore the FASTA software. FASTA usually stands for Fast or Fast A. It was a software which was being first developed even before the BLAST, and it is the first database similarity search tool developed in 1985 by Lipman and Pearson, and this was a software which was being first developed for the similarity search of proteins by the EMBL database, but later on it was also being extended to the nucleotides. FASTA is another sequence alignment tool which is also being used for the similarity of sequences, just as we used in the case of BLAST also. It is a pairwise alignment also here, but here there is a slight difference because it takes a little more longer time over here, and in a particular sequence the FASTA usually takes small parts known as a K tuples, and a tuple can be of about 1 to 6 bases or anything, and it matches these K tuples of one particular sequence to another sequence. That is, every 1 to 6 residues of a particular given query sequence has been compared to 1 to 6 sequences of another sequence which has been present in the database. And at the end of the matching process, when it reaches a threshold value, what happens? It will produce its particular result. And usually in the case of protein sequences, it is that every 2 residues are being considered to be as a K tube, and in the 6 residues is mainly considered to be for the DNA sequences. And so, unlike the BLASTA software, here in the FASTA sequence, what happens? The query sequence is broken into sequence patterns or words called the K tupules, and the target sequence is searched for these K tupules in order to find the similarities between the two. And it has been found that because when you give a particular sequence itself, that has been cut into 1 to 6 residues, and these 1 to 6 residues are further searched against the database. So what happens? The time that you take for the FASTA search is being found to be greater considered to the normal BLAST. And you should notice one thing, as we mentioned earlier, the FASTA is mainly being provided by the EMBL database, and BLAST is mainly being provided by the NCBI database. It was been initially provided by the NCBI database, but of course you know that various other databases has also been giving us BLAST software right now. And BLAST as well as FASTA, they have some similarities because they both of them use the same principle of comparing protein and DNA sequences. First, they check for similarities of protein as well as DNA sequences against the databases. So they are bioinformatic tools which will help us in this particular surfaces, and they use a scoring strategy to make comparisons between the sequences. They will say either 98% similarity, 68% similarity, like that. Both of them will produce a scoring strategy. Now, how similar these two sequences are? Are they 10% or 20% or 90% similarity? That has been provided by both BLAST as well as in FASTA sequences, and they provide us some accurate results also. But they do have some differences also. Now, in the case of BLAST, it's defined as a basic local alignment search tool available in the NCBI database, as we had mentioned earlier. FASTA is a program available at the European Bioinformatics Institute that provides facility to search similar sequences to one's query sequences, and the speed of BLAST is being found to be faster compared to the FASTA. Though the term implies fast A, when you consider, you will think that that is faster, but that is comparatively slow when you compare it to BLAST. And the accuracy of the results, BLAST will give us much more accurate results than FASTA, and FASTA is less accurate, as we mentioned earlier. And coming to the suitability, closely matched sequences, BLAST is very accurate. But if you have dissimilar sequence, FASTA is a better software which can be used. And you can, the ability to modify, BLAST is modifiable according to the need, and FASTA is not much modifiable. That is, the options that you get to modify your search is being less in the case of FASTA. Now coming to the use, BLAST is much more versatile and it is widely used than FASTA, and FASTA is less versatile and comparatively has less use than that of a BLAST sequences. So I guess you got an understanding of what is the difference between BLAST as well as FASTA. Both of them do the same purpose of comparing your sequence, your query sequence with a database, and you can see that the difference is that BLAST has been found to be a little bit faster. Now I think I can just take you to a link of the FASTA or the result of the FASTA sequence for your reference, and thereafter, like, I think it's better that we go through the site and we'll see how the FASTA looks. Now let's explore how to do the FASTA. Now this has been mainly been provided by the European Bioinformatics Institute, and it has been linked to EMBL, and that's why I had mentioned earlier about the EMBL also. Okay, so the FASTA search engine has been provided here. It's just that I just gave a Google search on FASTA and I entered into the FASTA search. I entered into the website, and you can see here you can either do the FASTA of the particular protein, or you can take off the nucleotide, or if you want a genome. Based upon what query sequence you have, you can go to different databases. Now if I need to do now this is a the FASTA link to search a protein, and they have given various databases against which my particular protein will be sequenced or searched. UniProt is the most important site. UniProt as well as which has been provided by different institutes. The UniProt site is being searched, as well as patents, structures, protein databases, etc. are being searched if I go to search a particular protein. And if I go down, you can just paste the protein sequence, or you can take choose a particular file, and you can select your parameters and you can just submit it. Now let me just go to the next one. We will take a nucleotide right now, and in the nucleotide you have got different databases over here, ENA databases, like that, different databases. So that is a European nucleotide archives. So you just paste your particular DNA sequence. Now here I've got a sequence in my tip of my finger, so I just pasted it over there, and I'm just going to give, you have different options. Either you can click be notified by email, okay, and I'm giving a title S7, okay. So I will get the result later on, because some I told you in the case of FASTA, you will take every 6 residues, 1 to 6 residues, and you will search for the similarity search. So a gap analysis is being carried out in the case of FAST. So it will take some more time than that of the BLAST sequences. So you give a submit, and you will get your information over there. And so they say that your job is successfully submitted, and they will send me the results in my mail ID, or it is available for 7 days also. Or if I want to see it right now, I just have to click this, and you can see the job is currently running. Please be patient, and they will be in the same window itself, it will be appearing after it. And compared to BLAST itself, it is having it takes more time. And see, this is a same sequence I gave 5 minutes back, even now it is running. So compared to BLAST, the FASTA takes more time. And I'll just show you a result which I have already obtained just before. Okay, like I had run this search previously, and the result what I got, I'm just loading that for your reference. We'll just have a look on that. See, you can see this is the previous result which I had got for the same nucleotide I had given in the database, and I can see that it is having now here. Just as I told you earlier, you get scores as well as the percentage identity is also been obtained, and it is saying that 98% similarity with pseudomonas aeruginosa are here, and you go down almost 98.2 with different databases, that is different sequences which have been used, that is available, and you can get a tool output. Different options are there. What are the tool outputs? You can see how frequently they are being exposed, and see here there is a gap also, that is, even they have put a gap in between this, saying that one is being, so you can find out mismatches between here. Okay, there is a gap which has been provided here because some particular sequences are not been present in this. They have put a gap in between that. In the case of FASTA sequence, on the FASTA analysis, you can find that. And see over here also they have put a gap, saying that so much regions are being found to be different. Like they did a pairwise alignment, and once there is some sequences which are not found there, they have left some gaps over there, and that's what in the case of, and they give you a percentage similarity also, and you can see the visual output somewhat similar like this. But the thing that you should notice, this was something which I did previously, and I got the result after many hours. So the case of FASTA, it takes more time compared to the other one, and compared to the, you also have links like what are the submission details, what all sequences I gave, that is all been provided here. And similarly, now you also have the protein, you have the link to a particular protein also. Now you just look, this one is a protein sequence, but I am going to put another sequence. You know, this is a nucleotide sequence. I am just also going to put here, I told you the dissimilar sequences also can be analyzed by FASTA. Okay, so if I go to put this here, I need to take what here, this, not protein, I need to take nucleotide itself, not the, because it is a nucleotide sequence over here, I need to paste this thing. Now in this particular sequence, now if I want to put a protein here, I need to put protein here, okay, and I need to place a protein sequence over here. So I am just pasting the protein sequence over here, and here again I give submit. I didn't give the information of about submitted by my mail, so I need to wait here until I get my results, or even if you want, you can just bookmark this particular page. No, yeah, I got the result very soon in the case of protein fast, but in the DNA it was taking more time. Now here it is saying that it is having high 100%. That's what we should note. This was a software which was initially developed for protein, so it runs faster in the case of protein compared to nucleotide, and the 100% similarity with thioredoxin OS methanothermobacter thermoautotrophicus, that one's particular protein, it was being obtained by various techniques and all that. So I guess you have an idea about how you get the FASTA analysis. In the case of proteins, it has been found to be, I think it's a little bit faster compared to the nucleotide sequences when you do a FASTA analysis.